All right. Well, hello, everyone. I think it's evening where everyone else is. It's just after 10 p.m. here in Toronto. So you might be wondering why I'm hunkered in my garage, just because the kids I hope are trying to go to bed now. And so it's a pleasure still to be here to share with you some of the research that we've been doing at the University of Toronto in the field of precision nutrition, where we aim to see whether or not we can use genetic testing to understand why some people respond differently from others to the same foods, beverages, and supplements that they consume. So most of the research that we've done over the past just over 20 years now, has focused mainly on cardiometabolic disease, but we've done work in other areas as well, looking at rheumatoid arthritis, work on breast cancer with one of my collaborators, Dr. Stephen Narod, who I'm sure many of you have heard of, one of the co-discoverers of BRCA1 and BRCA2. And so what we try to do is really look at optimizing nutritional status. And when we do this, that really impacts the health of individual cells in the body. And these are virtually all cells. So whether it's a cell lining the blood vessel that has implications for heart disease, or whether it's a mammary epithelial cell that has implications for breast cancer, or a kidney cell that has implications for kidney disease. Optimizing nutritional status of cellular health has implications for virtually all chronic diseases. So while most of the examples that I'll be giving today aren't specifically related to cancer as an outcome, it certainly has direct implications for it. A classic example, of course, is the work that we've done looking at obesity and weight loss, which we know obesity is an important risk factor for a number of different types of cancer. So in addition to also being a professor at the University of Toronto, I'm also the founder and serve as chief science officer of the company Nutrigenomics with an X, which provides genetic testing for personalized nutrition only through healthcare professionals. We do have a number of in-house dietitians that do provide consults, and oftentimes when I give talks to various groups, we like to provide a kind of a promo code for those who might be interested in that. So at the end, I'll have a QR code with a promo code for anyone who's interested in learning more about getting genetic testing for themselves to determine how to optimize their nutritional status. So first of all, the science of nutrigenomics, spelt ICS at the end, is a branch of nutritional science that, as you can gather by the name, deals with the interface between nutrition and our genetic makeup. And as I mentioned earlier, really trying to understand why some people respond differently from others. Now, from a research perspective, we can think of two sides of a coin. On the one hand, we know that diet can affect our genes, right? Nutrients can turn on and turn off various genes. And if we know the biological function of those genes, then perhaps we can tailor diet to alter the expression of those genes in a desired manner. Now, the flip side of that, and really the focus of our research program, and where I think the actual practical applications lie, are illustrated by that bottom arrow. And that's where we try to understand how variations in genes affect the way we respond to diet. So genes code for proteins that affect the absorption, the distribution, and the elimination of virtually everything that we ingest. Now, another element of the bottom arrow is before a gene can even act on a dietary factor, genes can influence the foods that we select. And this is an area that we stumbled across by chance a few years ago, where we were trying to understand why some people metabolize glucose and carbohydrates differently. Some people who have consume the same amount of carbohydrate, their blood glucose response is quite different. And what we found was this gene that we focused on, which was a glucose transporter, even though it didn't affect how people metabolized glucose, it was associated with habitual sugar consumption. So it turns out individuals who have a particular version of this gene just consume more sugar on a daily basis. Now, we know that sugar is a risk factor for things like type 2 diabetes. There have been some population-based studies and animal studies suggesting that sugar might also increase the risk of breast and colon cancer. So what's interesting here is we identified for the very first time that our genes can influence the foods that we select. And it was the kind of thing that got quite a bit of media attention. This was work that was done by a former PhD student of mine, Karen Enney. She is also a registered dietitian, and she happens to be the director of clinical services now for the company Nutrigenomics. But this was this was an article that appeared on the front page of the Toronto Star. The headline was "Craving Something Sweet, Blame It on Your DNA." A little bit sensationalist, not quite the headline we'd hoped for, but nevertheless, I mean, I suppose this is what sells newspapers. But importantly, it highlights and communicates to the public that our genes do influence to some extent the foods that we select or the foods that we avoid, and how much we tend to crave and how much we choose to consume. It doesn't mean that we can't override those genetic drivers, but it's important to recognize that they exist. And some people just have a much stronger craving for sugars in this particular case than others. And by being made aware of that, that can empower an individual. I myself happen to have that genetic variant, and I know that come 2 or 3 in the afternoon and I'm craving something sweet, I make sure I don't have like a large chocolate bar or something, because I'm probably going to polish it off. And when I have that craving, I recognize this is my glucose transporter talking now. You can choose a healthier alternative, something like a whole fruit that has other nutritive values, as opposed to some processed candy bar, just to get that same satiating effect. Now, when we look a bit more broadly at the science of nutrigenomics and why I think genetics is absolutely critical for us to understand the role of nutrition and any health outcome, whether we're studying breast cancer or heart disease or diabetes, if there have been enough observational studies, we tend to see this heterogeneity of response, right? These inconsistencies. One day we hear a study comes out and tells us that coffee is good for us. Another day, a study will come out and say coffee is bad for us. Another day, a study comes out and says coffee doesn't do anything. Well, there's a number of reasons for those inconsistencies. And an important consideration is the genetics of the population or the individuals being studied. So I try to kind of illustrate that in a black box. If we can identify an individual's genetic makeup or genotype, as we can describe it, then perhaps we can figure out who coffee might improve their health outcome. For some people, it might do nothing. But what about cases where the advice that we're giving actually causes harm? And if there's a way of identifying those individuals, finding alternate strategies, that's obviously something that we want to do. So again, I mean, you pick your nutritional factor and you pick your health outcome. If there have been enough observational studies, we know that findings are generally inconsistent. This was highlighted quite nicely just a few months ago in this article that was published just in November in JAMA, Journal of the American Medical Association, very high impact journal. And what this study looked at is the effects of sodium reduction on blood pressure. And it's important here because it highlights just how variable the response is. And these graphs that you're seeing in front of you here, these 4 graphs, are called waterfall plots. And the reason why I love these graphs is because they don't just compare group averages, they show the result of each individual subject in this study. And before I get into this in great detail, I just want to let you know that the conclusion of this study was that reducing dietary sodium intake results in a significant reduction in blood pressure. And the headlines said just that, right? This was the press release by the American Heart Association. And they said that reducing sodium intake significantly lowered blood pressure in as little as 1 week. But when you go back to the graph and you just look at the top left, and this looks at the changes in systolic blood pressure. Right? So that's that number that goes on top. You have like something like 120/80 as a good blood pressure. So 120 is the systolic blood pressure. Each line here represents the change in blood pressure. So the ones that are all the way to the left that go above the 0, that shows the biggest change in blood pressure. It's actually a drop in blood pressure. I know it seems kind of counterintuitive. I probably would have put those lines towards the left facing down. So what you can see here is roughly 74% showed a reduction in blood pressure, but just over 20% actually showed an increase in blood pressure. And if you look at the graph just towards the right there, that's the diastolic blood pressure. Almost 30% had an increase in blood pressure when their sodium was reduced. So that's the complete opposite effect. Now, we didn't see that in the headline, and that wasn't a conclusion by the researchers themselves, because what they do is they just compare the average in those who were on the sodium reduction versus those who were in the control group. And obviously, 74% is going to kind of predominate. That's going to overshadow the effect of the mere 20 or 30% that have the opposite effect. But what if you're one of those people, right? We used to call them outliers, try to figure out how to get rid of them from our statistical analysis because it just didn't look nice. It impacted our data. But what if you're one of those outliers where the advice that we're giving actually causes harm? And if there's a way of identifying that and finding something else, that's obviously something that we want to consider. So going back to this figure again, clearly one size does not fit all. And this is a concept that we've known for a very long time. In fact, one of the earliest references comes from the 1st century BC, where Lucretius noted that one man's food is another man's poison. What's good for one person might actually be harmful to somebody else. Well, we've come a long way, and with modern developments in molecular genetics and genomics, we can now pinpoint specific regions in our genome that can help us understand this. As you recall from high school biology, most of us, I should say, have 23 pairs of chromosomes, and on those chromosomes are stretches of DNA. And then we have our genetic sequence, the nucleotides, the ACGT, that make up the 3 billion base pairs of the human genome. And across this, our genome, we have alterations in the sequence from one person to the next. And these common genetic variants that we often call SNPs, which is short for single nucleotide polymorphism, they can explain why some of us look and function differently, why some people have straight hair versus curly hair, or brown eyes versus green eyes. So you can imagine if these single letter alterations can have such profound effects on how we look and function on the outside, imagine what they could be doing to us metabolically. Now, whenever these alterations are quite rare, some of them may actually cause diseases, right? And so when we think of some mutations or these rare genetic variants that occur in genes like BRCA1 or BRCA2, that actually is an important causal factor of breast and ovarian cancer, and perhaps some other cancer sites as well. But when we look at the other ones that have metabolic effects, while they don't actually cause any disease, they actually affect the way we metabolize nutrients that ultimately can impact our risk of various disease. So when we look to see where and how these genetic variants can impact our response to nutrition, as I mentioned, genes can influence our likes and dislikes of various foods. That ultimately determines what we choose to consume. Genes can also influence our appetite. Some people need more of a particular macronutrient to get the same satiating effect. And once a food is ingested, there's all kinds of genes that impact the metabolism, the absorption, the distribution, and the elimination of virtually everything that we ingest. Now, I just want to make a clear distinction between the different kinds of genetic tests. So unlike the disease-causing genes like BRCA1 or BRCA2, what we're looking at here are modifier genes. And these are genes that alone do not predict the risk of disease, but rather they modify the effects of environment. So as we all know, several years ago when Angelina Jolie tested positive and decided to undergo a bilateral prophylactic mastectomy, that really popularized genetic testing. And many women went out to get tested. And if you test positive, that means something. And there are things that can be done, including, things like surgical procedures like Angelina Jolie did. But if you test negative, what does that really mean? We all know, of course, that the vast majority of cases of breast cancer are not due to BRCA1 or BRCA2. So a negative test here, to some extent, and this might be controversial, but to some extent is a bit of a false assurance. Because again, most cases of breast cancer are not due to those particular genetic variants. So BRCA1 and BRCA2 associated breast cancer are really considered rare forms of breast cancer, relatively. And so genetic testing has been successful really for those rare forms of disease. But when it comes to the common forms of disease, they have not been successful because genetics alone do not predict the majority of cases of breast cancer and the majority of cases of heart disease or the majority of cases of diabetes. And so where the opportunities lie are really in the modifier genes for most forms of chronic disease. And again, these are the ones that affect the way we metabolize various environmental factors, not just nutrients, but even things like toxicants, environmental pollutants, and also medication. So the whole field of pharmacogenetics and pharmacogenomics help us understand how drugs can impact an individual, whether they're therapeutic, whether they have no effect, or whether an adverse drug reaction might occur in a particular individual. And this is really what provides actionable information where you can actually make specific recommendations. So back in 2007, I was presenting at the first International Nutrigenomics Conference in California. And there was a reporter there who wrote an article, and the headline was "Genetics May Define Diets of the Future," as though this was something that perhaps 30 or 40 years from now we would be doing. Well, it turns out it's not that futuristic after all. And this is evidenced by newborn screening programs. So in Ontario, certainly BC, and just about every province, and most jurisdictions in Western countries, every infant that's born undergoes a battery of tests for what we used to call genetic disorders. But it turns out the vast majority of these are actually managed by dietary means. So one classic example of an inborn error of metabolism is PKU, which is an inborn error in the metabolism of phenylalanine, which is an amino acid that's relatively innocuous, except in infants who have an impaired ability to metabolize it. And so it builds up to toxic levels and causes severe neurological dysfunction. Well, any product that contains aspartame, which is a dipeptide, meaning it's made up of two amino acids and one of them is phenylalanine, has to be labeled and it has to have that warning label. And so this is an example of a food product that has been labeled for a genetic subset of the population. So again, the idea that we'll be labeling food products as being futuristic is not really the case because we're already doing it, and this is just one example. Now, fortunately, inborn errors of metabolism are relatively rare, even though the phenotype is actually quite severe. But thinking of something that is much more common, even though it doesn't lead to what we would consider kind of a disease, is lactose intolerance. And there's a growing number of products now on the supermarket shelves that are labeled lactose free. And guess what? This labeling is essentially for those who have a specific genetic variant in the lactase gene. And that prevents them from being able to metabolize lactose. And then that causes gastrointestinal discomfort. So gas, diarrhea, bloating, stomach cramps, and symptoms like that. Now, most people don't need a genetic test to know that they have lactose intolerance. I say most because some do actually have it, but they're just not aware. Some people can actually push their microbiome to be able to metabolize lactose and are able to tolerate a little bit more. But by and large, anyone who has this genetic variant, if they drink a full glass of milk, they will definitely experience severe gastrointestinal discomfort, and that tells them that they do, in fact, have lactose intolerance. Now, I just want to go through and give some examples of, again, some various chronic conditions and cardiovascular disease, which is still currently the #1 cause of death in both men and women. So it's important for us to recognize what some of those risk factors are for cardiovascular disease. And coffee has been studied quite extensively in terms of its link with cardiovascular disease. There have been dozens and dozens of studies, and not surprisingly, the results have been all over the map. We, of course, were interested in trying to figure out if we can identify genes that can help us predict if coffee is good for us or bad for us. Now, when we look at a cup of coffee, it's a fairly complex beverage, all kinds of bioactives. Things like the polyphenols that have antioxidant properties might actually be beneficial. Other things like the diterpenoids that are known to raise LDL cholesterol, that's the bad cholesterol, not so good. So it's a mixture of some good things and not so good things. We asked the question specifically, is caffeine a component in coffee that might trigger a heart attack? This is just the chemical structure of caffeine. It's the most widely consumed stimulant in the world. Many people can't get their day started without a cup of coffee. When you look to see how caffeine is metabolized, it's broken down almost exclusively by the CYP1A2 enzyme. So this is an enzyme that is found mainly in the liver, and it converts caffeine into a more water-soluble substance called paraxanthine. Then that's more rapidly broken down into other metabolites and eliminated from the system. Now, the nice thing about the gene that codes for that enzyme is there's a common polymorphism or a genetic variant that has a profound effect on enzyme activity or inducibility, as you can see here on the y-axis. So as we know, we inherit one copy of most genes from our mother and one from our father. And so if both parents gave us the A allele or the A version of that gene, that effectively makes us a fast metabolizer. We have about a 4-fold higher rate of breaking down and eliminating caffeine compared to someone who has even a single copy of the C version, which makes them a slow metabolizer. Now, we reasoned that if caffeine is a component in coffee that might trigger a heart attack, then we would expect the slow metabolizers to be at a higher risk. right? Because they're less able to get rid of it. If it's going to have any adverse effects, it's going to be in those individuals who are not able to get rid of it from their system. Now, initially, we thought that the slow metabolizers just drank less caffeine, right? Because if it sticks around in their system longer, they don't need that second or third cup to maintain their buzz. But it turns out there was no difference in how much caffeine the fast and slow metabolizers consumed. And in hindsight, that's not really surprising because what you feel when you consume a caffeinated beverage are the stimulating effects of caffeine when it binds to the adenosine receptor in the central nervous system. Okay? You cannot just feel how much is floating around in your blood. And this is important because oftentimes when I present this work, someone will come up to me and say, oh, I'm definitely a slow metabolizer because if I have a cup of coffee after 1 in the afternoon, it keeps me up all night. Well, that doesn't mean you're a slow metabolizer. It just means caffeine binds more tightly to those receptors in the central nervous system. You cannot feel just how much is floating around in your blood. So before taking genetics into account, when we look to see the link between coffee consumption and the risk of a myocardial infarction, a heart attack, we found that those who drank 4 or more cups a day had a 36% increased risk of a heart attack. Now, the real question we wanted to ask was, is this increased risk due to caffeine? And if it is due to caffeine, we would expect the slow metabolizers to be at a higher risk. So when we stratified our population by fast and slow metabolizers, that's exactly what we found. On the left-hand side are the fast metabolizers, where you can see now moderate consumption is actually protective. So those who drank 1 or 2 to 3 cups a day had a significantly lower risk of a heart attack. Whereas the slow metabolizers, now as little as 2 to 3 cups a day, is associated with a significantly greater risk. And those who drank 4 or more cups a day had almost a 4-fold higher risk of a heart attack. That's about the equivalent risk of a pack-a-day smoker for heart disease. So we published this way back in 2006 in JAMA, Journal of the American Medical Association. And again, this was the kind of thing that got quite a bit of media attention. This was the front page of the London Times in the UK, why 2 cups of coffee can damage your heart. And the headline on the inside was, gene that could make your next coffee your last. Again, a bit sensationalist, but this is what sells newspapers. So it wasn't until a couple of years later that another group essentially replicated these findings. But rather than looking at risk of heart attack, they looked at the risk of hypertension, which we had proposed was kind of the intermediary pathway. And so even though the cup sizes are a little bit different and the groups are slightly different, nevertheless, you see a similar pattern. On the left-hand side, fast metabolizers are protected. On the right-hand side, slow metabolizers, the complete opposite effect. This has also been replicated in terms of prediabetes, looking at impaired fasting glucose, which we know is an important risk factor for breast cancer and as well as other forms of cancer like colon and prostate. And so, again, coffee increased the risk of impaired fasting glucose only in slow metabolizers, not in fast metabolizers. More recently, we looked at this effect in kidney disease, looking at kidney function. And this was also a study that got quite a bit of media attention. Some of you may have heard about this in the news just last year. We published this in January, and it was highlighted in February. This is just in Prevention magazine. This was also a study that we published in one of the JAMA journals, and this was work done by a former PhD student of mine, Sarah Madavi. She's currently the Director of Science and Innovation for the company. So what you can see here is the slow metabolizers on the left-hand side. The y-axis shows normal albuminuria. Now, this is a measure of protein in the urine. Now, normally, you don't want to have protein in the urine. So the kidneys should be filtering out proteins. But as we age and as our kidney function declines, we begin to secrete more and more protein. And so you can see that over time, this was a 16-year prospective study. You can see those with the light blue curve at the bottom there, that indicates a more rapid decline in kidney function compared to the slow metabolizers who consume less than 3 cups a day. But take a look at the fast metabolizers. There's actually 3 curves there. You can't even tell the difference. They're almost superimposable. And what that tells us is that among fast metabolizers, there's absolutely no effect of coffee on kidney function. We showed the same thing with another indicator of kidney function called hyperfiltration, and also with hypertension, because we know that the kidneys are very important in terms of regulating sodium homeostasis, which impacts blood pressure. And lastly, several years ago, this was a collaboration that we had with Dr. Stephen Narod and Joanne Katsopoulos, who's currently also a colleague of mine here at the University of Toronto. And we looked to see the association between fast and slow metabolizers among carriers of BRCA1. And interestingly, there was evidence to suggest that caffeine might actually be protective. And interestingly enough, we showed that in slow metabolizers, the protective effect of coffee seemed to actually be greater. And so it's important then to recognize, how do we balance the benefits of one condition like heart disease, as well as hypertension and prediabetes and kidney disease, with BRCA1 associated breast cancer. Now, this was a relatively small study, only about 170 cases and 240 controls, whereas the cardiovascular disease study, we had over 2,000 cases and over 2,000 controls. And likewise with the hypertension, much larger sample sizes. So, this has not yet been replicated specifically, but something to keep in mind. Now, shifting gears a little bit and talking about another important risk factor of breast cancer, which is obesity, and the whole area of weight management. Now, here we're not trying to use genetics to predict obesity. There's no point. There have been hundreds of genetic markers that have been linked to increases in waist circumference, adiposity, BMI, and other obesity-related traits. Here, what we're trying to say is for someone who wants to lose weight or feels they need to lose weight, can we use genetics to determine the optimal diet? There's all kinds of fad diets, as you know, low fat, low carb, keto, paleo, intermittent fasting, the list goes on. And the reason these fad diets persist is because they actually work in some people. So the question is, how do we know if that's going to work for me? Well, some of the most compelling evidence comes from Luke Hughes' group at Harvard, where they conducted this 2-year randomized controlled clinical trial comparing the effects of a low protein diet to a high protein diet. And as some of you may know, a high protein diet is very common in a lot of popular weight reducing diets. And here the effects of a high protein diet here really depend on which version of the FTO gene a person has. And this is looking at change or reduction in body fat, percent body fat composition, as shown here on the y-axis. So if you have either the TT or TA version of that gene, there's really no difference in how much body fat you lose, whether you're on a low or high protein diet. In fact, it looks like those on the low protein diet lost a little bit more body fat, but it's not statistically different. Now take a look at those on the right-hand side, those with the AA genotype. Those individuals who were on a high protein diet lost a considerably greater amount of body fat, and they maintained it 2 years later. This has also been replicated by a number of research groups, including us. We did this in a multi-ethnic population in Toronto. So just in the interest of time, I won't show all of that evidence of replication. But one question that often comes up is what happens when you give people their genetic information for precision nutrition? What do they do with that? Well, on the one hand, a person might say, you know what, I have that gene, it's going to motivate me to eat a healthier diet. But the flip side could be this concept of genetic determinism or genetic fatalism, where some people might say, you know what, it's all in my genes, there's nothing I can do, pass me the potato chips and remote and leave me alone. Now, this concept really applies more to those disease causing genes, where in some instances, there might not be very much a person can do. We know that that's the case in some conditions like Alzheimer's disease or ALS, where there currently aren't any treatment options available. But when it comes to the modifier genes, there is something you can do, right? If you tell someone you have the gene for salt sensitive hypertension, not just generalized hypertension, but salt sensitive, you need to lower your sodium intake. That's something that they can do, right? If you tell someone you're a slow metabolizer for caffeine, you need to limit your intake, that is something they can do. So we decided to put this to the test, and doing so using the gold standard randomized controlled clinical trial, where we compared the effects of giving people either DNA-based dietary advice that's tailored specifically to their genetic makeup or generalized recommendations for the population. And we found that those who got the DNA-based dietary advice actually understood the recommendations more, they wanted to learn more, they were more motivated to change their eating habits, and they actually changed their eating habits. They were more likely to comply with the recommendations, and they were the ones that actually needed to change the most. This has also been replicated by this was the first study of its kind. This was done by another former PhD student, Diva Nielsen. She's currently a professor at McGill University in Montreal, doing some really exciting research in the field of genetics, brain imaging, and eating habits. So that study also got quite a bit of media attention because, again, it showed for the first time that giving people the right kind of genetic information can actually motivate them to change their eating habits and actually result in more meaningful changes. This is another group at the University of Western that actually replicated this and expanded. Not only did they show improvements in compliance, but actually in outcomes. In this case, looking at changes in body fat. So for individuals compared to the control group, that's called the Diabetes Prevention Program, that's the DPP, those who also got a genetic test, and it happened to be specifically our test, the Nutrigenomics test. We had no involvement in this study. We had no control over the design or whether or not the findings would be published. They just used our test in their research. And fortunately, they showed that over and above the benefits of the diabetes prevention program, that a nutrigenomics test actually results in a greater reduction in body fat. So in terms of the benefits of genetic testing, I teach a fourth-year course on this, but in the interest of time, I really haven't been able to present all of the evidence, but these are just some: improved nutritional status, greater weight loss, improved compliance, greater motivation, and better understanding of the dietary advice. And lastly, also savings in healthcare for systems that actually incorporate genetic testing in usually for employee wellness programs. So some of the questions that often come up in this field is what type of practitioner offers this kind of test? The tests are not diagnostic, so mainly registered dietitians, but also physicians offer this kind of test. Information is confidential and it's blinded, anonymized. So we go to extreme measures to ensure patient privacy and confidentiality. Even though the test is not intended for children because the recommendations are not meant for them, many people do have their children tested. Most insurance does not cover the cost of the test, but for those who have insurance coverage for a registered dietitian, oftentimes some dietitians are able to bury the cost of the test in a precision nutrition package. And if you have coverage for a dietitian, that can actually not only offset the cost, but cover it entirely. And lastly, a question that often comes up is, who's a good candidate for a genetic test? And the answer, I think, is if you eat and drink and you care about your health, I think you're a good candidate. You don't have to wait until you're diagnosed with any kind of condition. Obviously, optimizing cellular health improves our overall health and reduces risk of just about every chronic disease. So if you recall that article back in 2007 talking about genetics defining diets of the future, fast forward 10 years later, and this article in the Wall Street Journal talked about not only our research at the university, but also the company Nutrigenomics, and said, this is no longer something futuristic. The science is here, the evidence is here. For those who do have access to that kind of information, that's something that they might want to consider. So I will stop here. Thank you for your attention. This is my email address. I will happily look at questions now and answer questions, but in the event that someone is watching this as a recording and hasn't had an opportunity to ask a question, feel free to send me an email. And as I promised earlier, this is a QR code for anyone who's interested in a nutrigenomics test, and there's a promo code there called NGX100 that takes $100 off the cost of the test, regularly around $400, and it brings it down to $300. So I will stop here and look to, I guess, the chat box, or if anyone wants to unmute their mic.